and also on the neuromuscular junction, which controls all of the muscle contraction. We're going to talk about the sliding filament theory first, and then we'll get into how everything is controlled. So, last time we talked about actin and myosin, and the thin filaments are the actin filaments. And so, why they call it sliding filament, if you can remember, actin and myosin are myofilaments. Myo just means muscle, and they're filaments, protein filaments. And so, when we're talking about the sliding filament theory, the thin filaments slide past the thick filaments because when a muscle contracts, it shortens. That's how a muscle actually contracts. In the relaxed state, thin and thick filaments overlap slightly. And we saw that when we were looking at the sarcomere. But upon stimulation of the muscle, the myosin head actually is going to bind to actin, and it pulls the actin. So the actin is going to slide, and the myosin is doing the pulling. The myosin does not move. Each myosin head is going to bind and attach several times, and they don't all bond at the same time. If they were to all bond and contract at the same time and let go, then you would have one powerful contraction, and then it would just let go and snap back into place. You wouldn't be able to hold any sort of contraction. And when this happens, sarcomeres are shortening. The sarcomeres shorten, myosin stays where it's at, and the actin is what is actually sliding. Now, in order for a muscle to contract, we have to have a nerve ending that is going to be controlling it. And we're going to be doing a couple of things on the internet where you guys are going to be seeing how this works. So it's going to make a little bit more sense once you guys do these little activities. But what the nerve ending is doing, it is propagating an electrical current. So neurons are going to be sending an electrical message or action potential along the sarcolemma. And the sarcolemma is the cell membrane of a muscle cell. And then we're going to have calcium actually come in to help with the contraction. Now, skeletal muscles are stimulated by a motor neuron. Motor neurons are neurons that are going to cause some sort of action of the somatic nervous system. Somatic nervous system is part of the nervous system that is voluntarily controlled. And the axons, remember, the axon is taking the message away from the cell and it's passing it to the muscle cell. So axons of motor neurons are going to branch, and they branch profusely so that they can get a message all the way through that muscle cell. And what this forms is something called a neuromuscular junction. And the neuromuscular junction is what is doing all of the controlling of the muscle contraction. So the neuromuscular junction is formed from the axon endings, and they almost come into contact with the sarcolemma. And when they come into contact, there's just a little bit of a space between the muscle and the neuron, but the axonal endings on the nerve side have something called synaptic vesicles. And these synaptic vesicles contain neurotransmitters. That's how neurons communicate with either organs or other neurons. In the case of muscles, the neurotransmitter is acetylcholine. And acetylcholine is the neurotransmitter that is specific to muscle contraction. Now, when the axon and the sarcolemma, or the cell membrane of the muscle fiber, come together, they form something called the motor end plate. And the motor end plate is on the muscle side, and it is a specific part of the cell membrane or sarcolemma that is going to receive, so it has these little receptors that are going to receive the neurotransmitter acetylcholine. And like I said before, on the neuron, the axon side, and then the cell side, the fiber side, the axon never touches the cell itself. So axons never touch anything. There's always a little space in between called a synaptic cleft. So this is what a neuromuscular junction looks like. So you can see this is the axon and it branches. This is the muscle fiber here. So this is the actual skeletal muscle. And as the axon branches, it forms neuromuscular junctions. So in this case, it's forming 3 neuromuscular junctions. And then this is a close-up version of this neuromuscular junction here. So this is the axon. And you can see this portion is the cell membrane or sarcolemma. Where the axon and the sarcolemma come into contact, this is called the motor end plate. So this is the axon or the nerve. These are those synaptic vesicles that have the acetylcholine. Acetylcholine is being released here via exocytosis and will be received by the motor end plate here. There's these little cups or receptors that are going to receive that acetylcholine. Now, once the nerve impulse reaches the end of the axon, there's going to be calcium channels that open inside the axon. We need calcium to have the neuron communicate, and then we also need calcium in order for the muscle to contract. So we need calcium for nervous impulses plus muscle contraction. And so the calcium is going to cause the vesicles to fuse with the axonal membrane, meaning that the synaptic vesicles will fuse to the membrane and then via exocytosis will dump the acetylcholine outside the neuron. So once the fusion, once the calcium allows for the acetylcholine synaptic vesicles to fuse, acetylcholine will be released via exocytosis. Once the acetylcholine is released outside the axon, then it's going to diffuse from high to low concentration across the synaptic cleft. And then there are these little cups or receptors on the sarcolemma, which is the cell membrane, to receive those. Once the acetylcholine binds to the sarcolemma on those receptors, then we can start the muscle contraction down the cell. Now, we don't want the muscle to continue to contract. One of the characteristics of skeletal muscle is that it is easily excitable and quickly excitable. It can contract quickly, but it also fatigues quickly. And so we don't want our muscles to contract and fatigue every single time. So what happens is the acetylcholine that's been received by the sarcolemma is going to be destroyed very quickly after it is received, so that we stop any further muscle contraction from happening. And there's always going to be an enzyme involved in order to destroy acetylcholine, and this is a really long name, but it is acetylcholine esterase. And the ASE always indicates that it is an enzyme. And so, like I said, it's going to be destroyed very quickly so that we don't have any further muscle contraction. So there's only one muscle contraction happening at any given time. Now, action potentials are a little confusing to understand, but we should remember that if something is polar, except for epithelial tissue, but if something is polar in science, it means that there are opposite charges on each side. And so you're going to see this again on the computer, and you're going to see this depolarization event, but it basically means that if we have a positive and a negative inside and outside of the cell, they're going to reverse. So we're going to end up having a reversal of these different charges in or outside. And as this happens, the depolarization is going to kind of run in one direction down the sarcolemma and then eventually down the T-tubule. Now, acetylcholine is very important. It's a neurotransmitter. We have many neurotransmitters that are in our bodies because each neuron is communicating with different organs and each other. But the specific neurotransmitter for muscle contraction is acetylcholine. And it's going to bind to the receptors, as I said before, on the motor end plate. That's the portion on the sarcolemma that has the receptors. And what's going to happen is they're going to, as soon as that's received, we end up having sodium and potassium diffuse. And when we have this occur, they're basically, the sodium is going to be coming inside the cell and the potassium is going to be leaving the cell. And I'm going to explain this further, and like I said, you guys are going to be able to see this happening in a little animation. So when this happens, we are going to depolarize the